Well, we're going to take a little bit of a different course of things just so that we make sure everybody has the opportunity to have all of the sessions. We'll be a little bit briefer on the next session, which is the Belzutifan session. So I'll ask Dr. Qian actually and Dr. Hammers and Dr. Brugarolas to come up. And then so that we can keep moving through the sessions for our specialty care session to follow, since some of our providers and our surgeons actually have surgery this afternoon and may have to leave a little bit early. What we will offer is don't tell anybody. You can bring your lunch in here and we'll have it sort of a eat through listening and talking. And those who need to decompress a little bit, feel free to have lunch in the cafeteria area, but we don't want to deprive anybody of any of the sessions today. So with that, on that note, I wanted to introduce Belzutifan. And we're talking about hypoxia inducible factor-2 inhibitor. That is Belzutifan, a first-in-class drug for the treatment of VHL-associated tumors. And again, here is the disclaimer and the disclosure of UT Southwestern's role in the development of Belzutifan and financial compensation that it has received from Merck and Company related to Belzutifan. So this is a story that took over a century to develop. Why is it VHL? Well, VHL is von Hippel-Lindau, and as with many things in medicine, this comes from the names of clinicians, providers. In the early 1900s, an ophthalmologist, Eugen von Hippel, described retinal hemangiomas, as Dr. Qian introduced to you earlier. Subsequently, a pathologist, Arvid Lindau, described the association between central nervous system hemangioblastomas and retinal angiomas, and subsequent identification was of more and more of the manifestations of what came to be known as von Hippel-Lindau Syndrome. We'll fast forward a little bit. That's describing, say 100 years of clinical and biological observation. And then there was a flurry of activity following the development of techniques that revolutionized study in the fields of molecular biology and biochemistry, of biological discoveries over a period of 20+ years that included the identification of the hypoxia inducible factor proteins. These are proteins that sense oxygen in the system, including the discovery of HIF-2, as Dr. Brugarolas mentioned earlier by Dr. McKnight and Dr. Russell here at UT Southwestern, the discovery of the von Hippel-Lindau tumor suppressor, the links that were established between VHL and regulation of the HIF proteins, and the identification that inhibition of HIF-2 was sufficient to suppress VHL defective tumor growth. I've just summarized just an extraordinary amount of work in 4 lines there. So it's not meant to minimize it. It's just meant to sort of provide an overview. As you heard earlier, this was a big deal. This biological connection is very elegant, and it led to the awarding of the Nobel Prize in Medicine and Physiology to Drs. Qian, Ratcliffe, and Semenza in 2019 for their discoveries of how cells sense and adapt to oxygen availability. And you can see the link there as well. Importantly, from a clinical and therapeutic perspective, this ultimately led to the FDA approval of this first-in-class therapy to target this interaction between HIF-2 and HIF-1 beta as a treatment for patients affected by VHL who have VHL-associated tumors and are not needing immediate surgery. And in 2023, the approval of Belzutifan for treatment of patients with advanced renal cell carcinoma, the preponderance of whom may not have VHL, but who have these sporadic mutations that lead to kidney cancers, following prior therapies that had been already established in the treatment of advanced or metastatic kidney cancer, including immunotherapies that are termed program death 1 or program death 1 ligand 1 inhibitors, and vascular endothelial growth factor tyrosine kinase inhibitors. So how do we get to this point? Now, Dr. Qian showed you earlier, so I can build on her slides, just this manifestation of this regulation of hypoxia inducible factors by VHL. So here in this cartoon, you can see depicted the HIF-2 alpha subunit, this HIF-2 alpha protein, under conditions of normal oxygen supply. So here we are in a cell, and this is sort of the cytoplasm of the cell, and here's the nucleus of the cell. And under conditions of normal oxygen supply, oxygen attaches to HIF-2 alpha on specific amino acids, or what we call residues, called prolines, as hydroxyl groups. That hydroxylation of HIF-2 causes it to be recognized by VHL, and VHL sends it to get destroyed. So that's how you lower the level of HIF-2 alpha protein to keep things under control. If there is not enough oxygen, you want HIF-2 alpha around. HIF-2 alpha can then interact with its partner, HIF-1 beta, in the nucleus of the cell. When these interact, this interaction enables this protein components to function as what's called a transcription factor, which Dr. Qian described earlier, to interact with hypoxia responsive elements on DNA to lead to the production of a number of signals that will support cell growth and, when deregulated, can cause tumors to form. Cell growth, survival, ability to spread or metastasis, and the feeding of the blood supply to provide nutrients to these cells or these tumors that should develop. So if you have VHL, you have a defective or inactive or lost VHL protein. And that loss leads to the constitutive activation of HIF-2 alpha with respect to its ability to signal these growth signals. This occurs in patients who have VHL syndrome, and it occurs in an estimated 95% of clear cell renal cell carcinomas that occur sporadically through somatic mutations. So over the years, the pathways that HIF and VHL signaling interacts with that promote cell growth and tumor formation and metastasis have been elucidated. And for a number of years, the identification of some of these targets, and Dr. Brugarolas described some of these earlier, led to the identification of things that you could try to use as therapeutic targets in the treatment of patients who have tumors that are caused by this inappropriate signaling. And most of this research was done in patients who had advanced kidney cancer, the preponderance of whom do not have VHL, but some of whom do. These are VHL-affected patients who develop advanced kidney cancer. Now, if you need to have a target for a therapy, there has to be something you can turn on or turn off to inhibit growth that is spurred on by that target. So early identification of targets involved the immune system and activating the immune system to affect advanced kidney cancer, but also these downstream targets of HIF. So one of the downstream targets leads to the production of vascular endothelial growth factors or platelet-derived growth factors to lead to signaling to vascular endothelial growth factor receptors and platelet-derived growth factor receptors on endothelial cells to promote blood cell formation and development to feed these tumors. These are enzymes. And so an enzyme you can turn off if you can get a drug to inhibit those enzymes. So the earliest treatments for these manifestations were targeting these enzymes. And here's a whole class of drugs that have FDA approval or had FDA approval for the treatment of patients with advanced kidney cancer by inhibiting these enzymes. There's also the use of antibodies that can target and block this VEGF. Similarly, as Dr. Brugarolas described, and he was involved in a lot of this work throughout his career, this interplay with mTOR and its signaling, and there are inhibitors of mTOR signaling for this growth. But all of these are associated with side effects, toxicities, as you might imagine. If you're inhibiting directly the things that cause the blood vessel formation to feed the tumors, that could have effects on blood pressure and normal blood vessels in your body. So ideally, it would be lovely to try to target the source. And if HIF-2 alpha is considered the source, if that would be sort of a canonical thing to try to imagine if we could inhibit that. Well, HIF-2 alpha is not an enzyme. You can't turn it on or off. And so through a lot of excellent work, again, that Dr. Brugarolas described earlier, from biochemists here at UT Southwestern, Rick Bruick and Kevin Gardner, and subsequently the development of Peloton Therapeutics, founded by Dr. McKnight, identified that you could inhibit this interaction between HIF-2 alpha and HIF-1 beta to prevent this interaction and its function as a transcription factor. So as this is all happening, this takes a lot of preclinical effort. You can't just come up with this drug and say, let's take it into patients. And I'm showing just two, simple examples here from a lot of work from two research efforts to take one of these early models of what became Belzutifan, this compound called PT2399, to test it in the preclinical setting. And you can see references from these articles from Dr. Brugarolas' lab and Dr. Kalin's lab showing the impact of treating tumors generated in mice that were kidney tumors with Belzutifan's precursor here, PT2399, and the decrease in tumor growth. You can see it represented here. You can see in this representation, tumors lighting up with this molecule, and looking at it through imaging, you can see the loss of tumor of this, again, this VHL-driven tumor. So I just wanted to turn to Dr. Brugarolas and sort of get your input. It's a big question and one that you could probably discuss for hours, but can you just describe, some of the efforts that it takes to before you ever go into human subjects and into patients, to test and vet with rigor these pathways and these possible new therapies to get to them to a point to say, okay, now we can bring that forward into patients.
Thank you, Kevin. So you just heard from Kevin, a very exciting journey going from the discovery of HIF-2 alpha to the development of Belzutifan. And just to be clear, Belzutifan and Welireg is the same thing. Now, what you may or may not know is that Belzutifan has another name, which is PT2977, and PT stands for Peloton Therapeutics. So this is a drug that was developed by Peloton, and then when Merck acquired Peloton, it changed the name to Belzutifan. So in the process of drug development, so this started with a chemical from our library. So UT Southwestern has a robotic facility, and they have several 100,000 chemicals that they were tested, looking for chemicals that were able to dissociate this HIF-2 alpha and HIF-1 beta partnership, right? And you need both of them for the HIF-2 complex to function. If you break them apart, they cannot work. And that provided a proof of principle. And then Peloton took those compounds and they went through hundreds and in fact thousands of iterations. That's why it's called PT2977, because they had a lot of different compounds that they were working through until they found one they thought was working optimally. Now, how do you know whether this is going to be helpful for patients? So traditionally, these chemicals are then tested on what we call our cell lines. What are cell lines? Well, you can take the cancer from a patient, and then you can ground it up and make a cell, and then you can adapt that cell to grow in plastic in what we call tissue culture. And then you can add the drug to the cells in tissue culture and see whether the drug has an effect on those cells. And in this case, would be cells from a kidney cancer. As it turns out, if you do that experiment, Belzutifan has minimal effects. And that's because when the cells are growing, when kidney cancer cells are growing on plastic, they don't need HIF-2. You can take those kidney cancer cells and you can put them into mice and you can do the same experiment. But of course, because those are just cells that were generated from a tumor and that they were passaged extensively in plastic, those cells have typically evolved and they are not a particularly robust system. So the program that we have at UT Southwestern is a program where we do the testing directly on patient cancers, but we do the testing directly on patients' cancers outside of the patient. So now for over a decade, we've been approaching patients that have surgeries, in particular if they have an aggressive kidney cancer, and we've asked them for permission. Would you be okay if we take the kidney tumor that the surgeon, like Dr. Margulis, is going to remove, and instead of throwing it into the trash, after the pathologist looks at it, we keep it and we use it for research. And we've in fact had over 1,300 patients that said, yes, we are happy for you to use our cancer. And then we take that cancer and we cut it into small pieces, and then those pieces we can transplant. We do surgery in mice. We take that cancer and we put it into the kidney of mice. And we don't add anything. We don't, the tumor goes from the patient in the OR to the mice in a matter of a few hours. And then the tumor grows in the mice. The mice have to be immunocompromised because if not, the mice would reject the cancer. And that way we can test the drugs that Peloton developed in the mice. And Peloton had a tool compound that is very similar to a drug that got approved. And we tested this drug in tumors from over 20 patients, including clear cell, and you heard from Dr. Qian, also the non-clear cell tumors. And we found that the drug was effective in about 50% of the kidney cancers that we had taken from patients. Now, these were kidney cancers from patients that have sporadic, not the familial VHL kind, which are actually harder to treat. Belzutifan is not as effective in those patients as it is in VHL patients. This was very encouraging because it showed that this drug by itself had activity. And in fact, we tested in tumors that had been from patients that had previously received other treatments and that the tumor was growing and the patient needed surgery. So we knew that even after previous treatment, this drug could work. And doing those tumors, we validated the mechanism of action. We showed that indeed the drug split the HIF-2 complex and that blocked the activation of the genes that are regulated by HIF. And unfortunately, we also found that resistance can develop, and we figured out the mechanism. But going back to Kevin's question, the process of drug development is a challenging process because you start with a chemical, but then you need to show that the chemical works for the intended application. And unless one has a program like we have over here, where we have access to patient tumor samples, if you rely on cell lines, the answers you can get may not be that great.
Thank you very much. And so, as you imagine, it's a huge effort to try to build us to this point. And so now we get to this point and you want to test this potential therapy in human subjects. And that's where we're indebted to patients, because this is, calling on volunteers. These are patients who volunteer and say, I'm affected by this condition, in this case, VHL, and I have a kidney tumor, and I am going to take part in a clinical trial and say, I will be a test subject. I will take this medication and we will see what happens and see if there's benefit to me or not, so that there may be benefit for others down the line. And so I'm showing you here the title from the published paper showing that the study of Belzutifan for renal cell carcinoma in patients affected by von Hippel-Lindau disease. So kind of kicking off this discussion, so we were fortunate, as Jim alluded to earlier, to have been involved with the early phase clinical trials leading to the development of Belzutifan here at UT Southwestern, and sorting out the strengths and the weaknesses of those drugs and things that needed to be altered or tested or vetted. Ultimately, Belzutifan as an agent was developed. So I will ask Dr. Hans Hammers, who many of you know. He's an internationally recognized expert in kidney cancer and has been involved in the development of standard of care therapies for patients with advanced kidney cancer. Dr. Hammers, if you can discuss sort of the process of executing these clinical trials and what's involved in terms of the testing safety and testing tolerability, and what kinds of things we look for in measuring activity.
Sure, absolutely. My pleasure. So, it's a long journey, right? From testing a chemical library to actually finally a product that can go safely into humans and hopefully have efficacy. It's a heavily regulated process, obviously, because it needs to be done well. And anything that touches patients, we need to make sure that we don't give them overt poison. So, part of the package, if you will, that needs to be developed before it actually ever gets into humans is actually extensive toxicity testing and validation in preclinical models, such as Dr. Brugarolas was mentioning, to make sure, A, there is a good rationale that this is actually a potential drug candidate, but also that the dog that they give it to before they give it to humans doesn't drop dead, right? So there actually goes extensive testing of some of these front runners of potential compounds in animals, mice, rats, dogs, sometimes additional animals. And then you also get a sense of, what's the dosing of these drugs that we need to have potentially an effect in these cancer cells? What's the drug level that I may need to achieve in humans? For example, you may get an initial impression where to maybe start with a safe dose. And then it typically goes into very early phase clinical trials where the initial patients will most likely start at a very low dose level, and then there will be a dose escalation. And, and again, we are grateful for the pioneers who participate in these clinical trials, right? Participating in the phase 1 clinical trial is probably something that initially everybody will look at with some form of anxiety. And gosh, this was never in humans, the first in human trial, right? This class of drug has never been in humans before. And so certain things that you may or may not expect, that can be what we call on-target effects or side effects, meaning the very mechanism that's being targeted can lead to anemia, for example, and other complications. But could there also be other targets being hit in humans that may give you, for example, nausea, vomiting, or other things? Fortunately, that's actually very little the case with Belzutifan. It's a very unique drug, and the primary side effects are really what we call on-target related. You may also start with a candidate that might not be the perfect candidate. And in fact, in patients, and that's actually how things started. The very first phase 1 clinical trial was actually led by Dr. Courtney right there with a compound called PT2385. I know we have all these different numbers, et cetera. And John Josie will know much better all the insights, how this drug was developed. But, but there was an insight in, hey, the metabolism isn't great, or, we don't really achieve the drug levels that we really need to have to be effective in patients. And then they looked at their portfolio of agents and said, hey, why don't we try this other one? This might be better suited, slightly different structure. And hey, that was then the winner. And then you go to dose escalations, see what is a potentially effective dose. Do you hit a certain biomarker, for example, with erythropoietin levels or others? And do you have essentially arrive at a certain dose level that's then recommended to go into more broader efficacy testing? So it's all about, what is safe to give, what is a reasonable drug level from toxicity and also efficacy perspective. And then you go into trials that will show efficacy to prove to the regulatory authorities that this is actually something worth paying for, that is effective in humans and safe. And then you go into efficacy clinical trials that can look very different depending on what you treat. If you're treating something that is more rare, such as von Hippel-Lindau syndrome, right? You may not have thousands of patients to work with like we have in sporadic kidney cancer, right? So many fewer patients, and you may have a different trial design and different outcome measures to convince the regulatory bodies than you would need, for example, for sporadic renal cell carcinoma, which limped behind, actually, quite frankly, the development for von Hippel-Lindau disease. So I think it's a huge success story in medicine. I remember the very first time that a patient was benefiting from it in my clinic. The patient was in tears because this is the first time there's something for me. So really, so kudos to the team, kudos to what happened here at this institution. So.
Thank you, Dr. Hammer. So this is just a few examples from that clinical trial, that efficacy clinical trial for patients affected by VHL. And for safety reasons, it was very specific. They were looking for patients affected by VHL who had renal cell carcinoma. These are patients who had renal cell carcinoma tumors that were no larger than 3 centimeters. And as Dr. Qian alluded to earlier, if you go above 3 centimeters, you want to have an intervention because the risk that it could advance and spread increases. The intention here was to see, can you arrest the growth? Could you keep these tumors at bay? Patients in this trial had, in many cases, many other VHL-associated tumors, and they captured those data as well. But the primary endpoints they were looking at to sort of have a really safe and rigorous testing was at the kidney tumors. And in this trial, there were 61 patients, excuse me, who enrolled, and they looked at responses. Did the tumors shrink? Did they get bigger, or did they stay the same size? So a complete response is the tumor goes away completely. A partial response, it shrinks by a measurable and substantial amount. Stable disease is it stays stable and it doesn't keep growing. And disease progression is that tumor keeps growing. And they assessed what was the best objective response among these patients who had kidney cancers with treatment with Belzutifan. And as you can see here, there were no complete responses, but there were also no patients who progressed. So this was basically either shrinking or keeping stable these tumors. This is what we call a waterfall plot. This is the change in the target tumor lesion on treatment with Belzutifan. So if you have more than a 30% change in the measurement of the tumor from baseline, that's called a partial response, and that's shown in green. And if you have less than 30% chance decrease and less than a 20% growth, that's stable disease. This is everyone on the trial. There was nobody going like this, and that's what we mostly see. Usually we'll see people going like this and more people going like this for a drug that comes into development. And I like to share this because I think this is very clinically meaningful for patients in the VHL community. This plot shows at this solid line at day 0 here. Day 0 is start of Belzutifan. These are years before Belzutifan, going back in some cases for patients 10 years. These are years after Belzutifan to the completion of the follow-up for when this trial was reported, which was a bit more than 2.5 years. Each of these purple dots is the number of procedures patients had to have to treat their tumors prior to starting treatment on the trial with Belzutifan. Each of the blue dots is the number of treatments, interventions these patients needed after starting Belzutifan. So you have to be paying a close attention to capture something like this, because this is life-changing, even if those tumors are still there. If they can stay there and stay stable and not cause problems, that's a big deal. Now, all of these treatments are going to be targeting this activity, and there may be side effects. So Dr. Qian, I wanted to get your perspective as somebody who sees many, many patients affected by VHL. Can you describe sort of your clinical experience, how, as you're treating patients with Belzutifan, how you monitor those patients for safety, how you monitor them for activity, and how you approach and address some of the side effects that are, in the case of Belzutifan, typically on-target side effects. It's doing its job, and so it might cause hypoxia, it might cause anemia. Can you share that with us?
So I will say Belzutifan is one of the very well tolerated medications that I have in my pocket for patients. And so some of the common on-target side effects is anemia or low red blood cells, right? Because of the pathway that the HIF pathway helps with, we certainly see anemia or low red blood cells in almost all patients. But for some patients, it's a little bit, they barely feel anything. And for some, it's a little bit more. So for most patients, we watch their blood count about once a month, and we look at that red blood cell count. If it's above 10, usually patients are okay, and we don't have to give additional intervention for it. Once it gets into the low 9s, I start thinking about maybe giving them a shot to help bring up the red blood cells. And almost never do I have to give transfusions because usually it does not go that low. And again, we try to catch it before it gets that low, we give a shot, and it usually helps us bring the red blood cells up. The other most common side effect is feeling tired. And so fatigue is quite common, and I see it in quite a few of my patients. Now, a lot of our patients are, well, I see two ends of the spectrum. Either I see the 40, 50-year-olds, right, who's been dealing with VHL for decades before we had this drug. And I'm starting those patients on Belzutifan with the hopes that I'm going to prevent them from further surgeries and procedures, right, that I can shrink their tumors to help them have a better quality of life. The other end of the spectrum that we're seeing a lot of now are the 19, 20-year-olds, right? That gets referred to us from pediatrics. They may have one or two hemangioblastomas. They may not have anything, or they may have a small, let's say, kidney tumor, right? So my job is to try and prevent these very young patients from getting surgeries to their brain, their spine, their kidneys, right? By giving them Belzutifan in the right indication. And so when we think about things like anemia and fatigue, we have to think about the patient as a whole, right? A lot of these patients are in college, right? They're in another city. How do we manage their side effects and look at their labs and their imaging when they live so far away from us, right? So I'm seeing a lot of patients on their fall break, right? Their, winter break, their summer breaks, right? So there's a lot of coordination that we do with the patient and their family members so that they can stay in school, right? So that they're not having to come in all the time for these tox checks at the during their school time. And so fatigue and anemia are usually the most common things that we hear. And then it could cause a little bit of liver irritation. And so we also look at the liver function, kidney function. I do about once a month for these patients as well. And then the most uncommon but dangerous one is what we call hypoxia or decrease in oxygenation. And so all of my patients, I tell them to buy a pause ox to keep at home. And especially when we first start the treatment, check it every day, right? If the oxygenation is starting to drop, that's an emergency. You call me and I will tell you what to do. The good thing about Belzutifan is when you stop the drug, the side effects kind of go away. And so they don't tend to be long-lasting side effects. They tend to recover when we hold the medication. And so our job is to see what the side effects are and to manage it with you, right? And so that might mean hold it for a little bit. That might mean decreasing the dose a little bit. And so we kind of manage it with the whole patient in mind, right? And their family in mind. So those are kind of the main things I see. Most of my patients are able to be at the full dose, and we manage the side effects. A few we have to dose reduce a little bit, but majority of them are actually able to tolerate the full dose of Belzutifan.
Thank you so much. This was terrific. To keep things moving forward so that your day doesn't get too long and so you don't miss your shuttle back as well, what we were going to do now, since we're running a bit over, is our panelists will be available if you have specific questions related to Belzutifan. I think a lot of these questions came up earlier, but we'll just take about 10 minutes for people to use the restroom and get lunch. And then, as I said, don't tell anybody. We'll break the rules and we'll carry forward with our next session because I think some of our surgeons have some cases they have to do this afternoon to go forward. Dr. Josie.